Welcome to the deep dive. Today, we're going to be exploring a concept that's really key to understanding how drugs work.
Okay.
Dose-response curves.
Yeah.
So whether you're prepping for a pharmacology review or just really curious about the science behind medications, we're going to break it all down for you.
Yeah, it's really fascinating how our bodies react to medications. It's not
Wow.
always a simple, more drug, more effect scenario.
Right.
And dose-response curves help us visualize this relationship.
Yeah. We've got a stack of sources here, some academic papers, even a YouTube video on cell signaling.
Oh, wow.
You even sent over some interesting articles about thyroid hormone receptors.
Yes.
Our mission today is to really break down these curves step by step.
Okay.
So you grasp not just what they are, but why they're so crucial in drug development.
Sounds good.
So let's start with a bit of an analogy here.
Okay.
Imagine a cell as a bustling city, right? With receptors on the cell surface acting as gates.
Mm-hmm.
Drugs are like messengers, each carrying a specific message to these gates.
I like that.
Okay, so different messengers can deliver different messages, even if they arrive at the same city gate.
Exactly. Think of heart muscle cells, for example.
Okay.
When messengers acting on beta-1 adrenergic receptors arrive, you get a strong, immediate response, increased heart contractions.
Yeah.
But other messengers, and even those activating similar pathways, like those acting on EP4 receptors, might not elicit the same response.
So how is that possible? How can two messengers activating similar pathways lead to different outcomes?
Yeah.
One thing that really caught my eye in these sources was this concept of microdomains.
Right. So microdomains are like specialized neighborhoods within our cellular city.
Okay.
They're areas where specific signaling molecules cluster,
Okay.
influencing how a message is received and interpreted.
So it's all about location, 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 even within a cell.
Precisely.
Wow.
And that brings us to dose-response curves. These curves visually represent how a drug's concentration relates to its effect.
Okay.
It's a graph with drug dose on the x-axis and response on the y-axis.
So like a map showing the impact of different messenger concentrations on our cellular city.
Exactly. And when we're looking at these curves, there are two key factors that stand out.
Okay.
Potency and efficacy.
Let's break those down.
Okay.
Potency first. What exactly does that tell us?
So potency indicates how much of a drug is needed to trigger a response.
Okay.
On the curve, a more potent drug acts at lower doses, shifting the curve to the left.
So potency is all about how much of the messenger is needed to get things going.
Right.
Yeah.
Now, efficacy is about the maximum effect a drug can achieve regardless of the dose.
Okay.
A drug with higher efficacy will have a taller curve, indicating a greater maximum response.
So potency is how much you need. Efficacy is the strength of the effect.
Exactly.
Got it.
Yeah.
I'm guessing, though, not all messengers are created equal. Right?
You're catching on. Okay.
So we classify drugs based on how they interact with those city gates. The receptors, full agonists, are like highly persuasive messengers. When they bind to a receptor, they unlock the full potential response.
So they get the city gates to swing wide open.
You could say that.
Right.
Their dose-response curve would be tall, reflecting that maximum response.
Gotcha.
Then we have partial agonists.
Okay.
They bind to the receptor, but only elicit a partial effect. Their curve might have a similar shape, but it will be shorter, indicating a lower maximum response.
Okay, so they can only nudge the city gate open a bit.
Yeah.
What about those messengers that try to shut things down entirely?
Those are the antagonists, the city guards.
Okay.
They block messengers from reaching the receptors,
Right.
preventing any response.
Gotcha.
Their curve would be flat since they're actively working against an effect.
Okay, so we have our curve, we understand potency and efficacy, and we know about our different types of messengers.
But how does this all connect to drug design and that sweet spot you mentioned earlier, the therapeutic window?
That's where things get really interesting.
Okay.
Dose-response curves are crucial for determining this therapeutic window, the range where the drug is effective but doesn't cause unacceptable side effects.
So it's about finding that Goldilocks zone where the drug is doing its job without causing too much trouble.
Exactly. Dose-response curves help scientists determine how much of a drug is needed to hit that sweet spot.
This is all starting to make sense now.
Good.
Those curves aren't just lines on a graph.
Right.
They're like blueprints for understanding a drug's potential.
Yeah.
But earlier you mentioned microdomains.
Yes.
How do those fit into this intricate cellular city?
Well, let's go back to those heart muscle cells with their beta-1 adrenergic and EP4 receptors.
Okay.
Remember, those different receptors lead to different responses, even when activating similar pathways.
Right.
Microdomains help explain why.
So those microdomains could be the key to understanding how these messengers cause such varied effects within the cell.
Precisely. Think of kinase anchoring proteins, or AKPs, as the city planners.
Okay.
They ensure that specific signaling molecules are concentrated in the right microdomains, like making sure the right messengers end up in the right neighborhoods.
So it's not just about the message, it's about delivering it to the right location within the cell.
Exactly.
Oh.
And the type of AKP present can influence which signaling molecules cluster in a specific microdomain.
Okay.
This spatial organization adds another layer of complexity to how a cell responds to a drug.
Wow. Okay. This is getting really fascinating. We've gone from cells as cities to messengers and gatekeepers, and now we're talking about city planners directing traffic within the cell.
It's amazing, isn't it?
It's incredible how much complexity there is in something as seemingly simple as a drug interacting with a cell.
Yeah,
So
it really is incredible.
I feel like we're only just scratching the surface here.
Yeah, we were just getting started. There's so much more to uncover about how these microdomains influence drug action, and we'll dive deeper into that in the next part.
Sounds good.
All right.
Okay. These microdomains are really making me rethink how we view drug interactions. It's not just about finding the right key for the lock.
Right.
It's about making sure that key ends up in the right place within the cell.
Yeah.
Those articles you shared about thyroid hormone receptors really highlighted that complexity.
You're hitting on a crucial point.
Yeah.
Those receptors are involved in so many vital processes,
Right.
from metabolism to brain development, and their activity is tightly controlled by thyroid hormone, or T3.
Right. And the articles mentioned how these receptors can actually silence gene expression when T3 is absent.
Yes.
Then when T3 binds, it's like flipping a switch.
Right.
They activate gene expression.
Exactly.
Well, it's not just about whether the messenger is there. It's about how it changes the receptor's behavior.
That's a great observation. And this is where dose-response curves become crucial for understanding thyroid hormone action.
Okay.
Researchers can use these curves to determine how much T3 is needed to activate those receptors and trigger specific gene expression patterns.
That makes me think about the different types of agonists we discussed earlier.
Right.
Would T3 be considered a full agonist in this scenario? Since it seems capable of fully activating the receptor and triggering the maximum response.
That's a sharp question, and it highlights the importance of context.
Okay.
While T3 might act as a full agonist in certain situations, it's not always that straightforward.
Okay.
The body is a complex system,
Right.
and multiple factors can influence a drug's effect.
So even with a full agonist like T3, there might be other molecules or pathways modulating the response.
Yeah.
Like there's a whole network of checks and balances within the cell.
Exactly. And that's why dose-response curves are so valuable. They allow us to dissect these complex interactions and visualize how varying doses of a drug like T3 affect the target cell's response.
So by understanding these dose-response relationships, scientists can develop drugs that target specific pathways with greater precision.
Absolutely.
And they can potentially predict side effects or interactions with other medications.
Absolutely.
Wow.
These principles apply not only to thyroid hormone, but to countless other drugs and signaling pathways.
Okay.
Dose-response curves provide a fundamental tool for understanding how drugs work at the cellular level.
It's amazing how much information is packed into those curves. They're like a secret code for unlocking a drug's potential.
Yeah.
But we can't forget about the therapeutic window,
Right.
that delicate balance between effectiveness and safety.
The therapeutic window is all about minimizing harm while maximizing benefit.
Right.
And dose-response curves are essential for pinpointing that sweet spot.
Okay.
By studying how different doses affect both target cells and other cells in the body, scientists can identify the range that delivers the best therapeutic outcome with the least risk.
It's like navigating a tightrope.
Yeah.
You need enough of the drug to reach the destination, but not so much that you lose your balance.
A perfect analogy.
Thanks.
And those articles about thyroid hormone receptors mentioned how mutations in these receptors can lead to serious health problems.
Right, problems like resistance to thyroid hormone or even cancer. It's like the
Yeah.
communication system within the cell gets scrambled.
Exactly.
Yeah.
And understanding how those mutations affect the dose-response relationship is crucial for developing effective treatments.
Okay.
For example, if a mutation makes the receptor less sensitive to T3, higher doses might be needed. But that also increases the risk of side effects.
So it's a constant balancing act,
Yeah.
trying to achieve the desired effect without tipping the scales too far in the other direction.
That's the challenge and the beauty of drug development. It requires a deep understanding of the intricate interplay between drugs, their targets, and the body's responses. Dose-response curves are essential tools in this endeavor.
They're like a roadmap guiding scientists towards the most effective and safe treatment approaches. It's fascinating how these seemingly simple curves can reveal so much about the complexities of drug action.
You've got it. They provide a framework for unraveling those complexities.
Yeah.
And with new technologies and research methods emerging all the time, our understanding of dose-response relationships is constantly expanding.
I'm starting to see those curves everywhere now. It's like they hold the key to unlocking a deeper understanding of how drugs interact with our bodies.
Yeah.
But I have a feeling we've only just begun to explore their full potential.
You're exactly right. There's still so much to discover. But for now, let's shift our focus back to those fascinating microdomains.
Yes, let's dive back into those cellular neighborhoods and see how they're shaping our understanding of drug action. So we've been talking about cells as cities, with messengers and gatekeepers and even city planners directing traffic.
Right.
These microdomains really highlight the complexity of what's happening inside our cells.
Yeah, they're like control centers.
Okay.
They fine-tune how signals are received and processed within a cell. And understanding how drugs interact with these microdomains is opening up new possibilities for drug development.
It's no longer just about finding a drug that binds to a specific receptor.
Right.
It's about making sure it reaches the right neighborhood within the cell.
Precisely. We're moving towards a more targeted approach, aiming for greater precision and effectiveness. And those kinase anchoring proteins, or AK-KIPs, play a crucial role in this.
Right, those AK-PIPs are cellular city planners.
Yes.
They act as scaffolds, bringing specific signaling molecules together within those microdomains.
Exactly.
It's like they're creating hubs where the right players can meet and interact.
Exactly. And because different types of PKA have different affinities for various ACAPs, you get incredible specificity in how signals are transmitted.
Okay.
It's like a finely tuned orchestra,
Right.
with each ACAP contributing to the overall harmony of the cellular response.
But what happens when that harmony is disrupted? Those articles about thyroid hormone receptors mention some serious consequences when things go wrong in those microdomains.
Yes, mutations in those receptors can disrupt their interactions with AK-PEPs and other signaling molecules.
Okay.
This can lead to abnormal cellular responses, like a conductor who's lost their baton.
Right.
The signals get mixed up.
Gotcha.
For instance, if a mutation prevents a thyroid hormone receptor from binding to its designated AK-PEG, it might end up in the wrong microdomain,
Okay.
triggering inappropriate gene expression.
So it's not just about the receptor being present.
Right.
It's about being in the right place and interacting with the right partners.
Yeah.
It's like a delicate dance.
Yeah.
And even a small misstep can throw everything off.
That's a great way to put it. And this highlights why understanding microdomain organization is so crucial for drug development.
Okay.
By targeting drugs to specific microdomains, we might be able to correct these signaling errors and restore normal cellular function.
So instead of just trying to block or activate receptors,
Right.
we're moving towards fine-tuning their activity within specific cellular compartments.
Precisely. It's a whole new frontier in pharmacology.
Wow.
And it's incredibly exciting. As we learn more about how these microdomains function, we can develop drugs that not only target specific receptors, but also modulate their interactions with AKPs and other signaling partners.
This could lead to treatments that are more effective and have fewer side effects.
Exactly.
It's like we're learning to speak the cell's language instead of just shouting commands.
That's a fantastic analogy.
Thanks.
And with advancements in imaging techniques and molecular biology tools, we're gaining an unprecedented view of the intricate workings within our cells.
Right.
This knowledge is revolutionizing how we approach drug discovery and development.
It's like we're finally starting to appreciate the complexity and beauty of this microscopic world. Those dose-response curves we talked about earlier, they're not just static graphs anymore.
Right.
They're dynamic representations of these intricate cellular interactions.
Well said. And I hope this deep dive has given you a new appreciation for the amazing world of cell signaling and the crucial role dose-response curves play in understanding it.
It certainly has. The next time I hear about a new drug or treatment, I'll be thinking about those curves, those microdomains, and all those fascinating interactions happening behind the scenes.
Remember, the more we understand about how drugs work, the better equipped we are to use them safely and effectively. Keep exploring, keep asking questions, and never stop being curious about the world around us.
Yeah.
There's always more to discover.
Thanks for joining us on this deep dive into the fascinating world of cells and drugs.
It was my pleasure. Until next time.